It is now my pleasure to turn today's program over to Patrick Lowe. Patrick, the floor is yours.
Welcome to today's webinar titled Exploring the Role of iPSC-Cardiomyocytes in Drug Discovery and Safety Assessment, brought to you by Biotechniques and sponsored by ACEA, Biosciences, and Cellular Dynamics International. Hello, my name is Patrick Lowe, and I am a senior editor with Biotechniques. In this webinar, our speakers will describe the use of human-induced pluripotent stem cell-derived cardiomyocytes and instruments that assess cardiomyocyte contractility, viability, and electrophysiology for the discovery of drugs that modulate the contractility of cardiomyocytes and for the assessment of the efficacy and safety of combinations of cardiac drugs. Following these presentations, there will be a question and answer session with the speakers. So please feel free to enter any questions into your webinar console throughout the presentations. Our first speaker is Dr. Kristina Green. Dr. Green is a Senior Director and Head of Cell Biology at MyoKardia, Inc. She will describe the use of cardio-ECR technology to screen both inhibitors and activators of contraction for their activity and safety, while providing insights into the effect of cardiomyocyte differentiation on compound activity. Kristina?
Okay, great. Thank you so much for the opportunity to talk about our experience with cardio-ECR in building our drug discovery capabilities utilizing iPSC-derived myocytes. Oops, I'm sorry, I need to advance to my slide. Our goals for the drug discovery program at MyoKardia is first and foremost to be able to find compounds which can provide therapeutic intervention for both hypertrophic and dilated cardiomyopathy by designing a screening platform that facilitates screens based on the desired phenotypic outcomes. Key to doing this is having relevant phenotypic models, robust assays with high signal-to-noise ratios, and a source of the relevant cell types. Here we have chosen to use iPSC-derived myocytes as a consistent and quality-controlled source of these cells. This will allow us to rapidly triage compounds prior to going into animal models for pharmacology and safety, as well as enabling studies of mechanism of action. Over the last 6 months, we've been building a more robust cell biology platform at MyoKardia. Early on, we had developed extremely useful high-throughput biochemical screens for activity and had very relevant and effective animal models. In our biochemical screens, we could screen through thousands of compounds. But in between our hit-to-lead screening, we had a very low throughput primary myocyte assay. This assay is very data rich and gives us insights into contractility with readings of amplitude, rates and velocities, and contraction and relaxation, calcium handling, but it's just not scalable. So it, in fact, created a bottleneck for us. What we really wanted is to develop a cell biology platform that would provide a testing funnel for us. To help take us from hits to leads, we wanted to build a testing funnel that would take compounds through increasingly complex studies that would begin with a higher throughput assay for effects of contraction and safety, then on to force measurements, and finally through into primary myocytes if necessary. We used our myocyte assay to inform what kind of information that we wanted to be able to acquire from our testing platform to include contractility data, contraction and relaxation velocities, off-target effects in terms of effects on sodium, calcium, potassium, or hERG channels, as well as information about hypertrophy and viability. Those would be excellent additions. As we set out to build our platform, we evaluated different technologies to decide what was truly fit for purpose in this situation. We've looked at microtissue technology, in which we can use iPSC-derived myocytes to form tissues attached to 2 posts to be able to directly evaluate contractility force in a loaded system. We also assess the utility of MEA devices that can only detect field potential. In our myocyte assay, we currently utilize a video capture system And we also started looking at other options which might add higher throughput to this primary cell assay. And importantly, we also evaluated the ACEA cardio-ECR, which can give us measurements of impedance and field potential. The cardio-ECR E-plates have wells that are the same size as a 96-well plate and are arrayed in a 48-well format. The bottom of each well, as you can see in the lower left panel, is mostly covered with electrodes that detect impedance. These electrodes have a very small current which is constantly running through them. When a cell sits on top of one of these electrodes, the current is impeded, and the equipment detects the impedance. The measurement is reported as an arbitrary number called the cell index. When myocytes derived from iPSCs are used in this system, they begin to beat spontaneously. So if you look at the right panel there, you can see that the beating, when cells contract, they have a different presence on top of the electrode than when they are relaxed. This beating can also be detected as changes in impedance. When the cells are sitting in a relaxed state, they have less impedance. When they are contracted, they have more impedance. This data generates, the data that's generated from the capture of these impedance readings looks like the readout from an EKG. Also, in each well, there are two field potential electrodes which can measure the action potential of the cells and show us where the sodium spike begins, as you can see there on the very right side of that graph, and then also where the T wave and repolarization begins, right after the reference point. So from the waveforms that are detected by the cardio-ECR, we can obtain measurements of contractility, which include contraction amplitude, beat rate, beat period, and contraction and relaxation times. The field potential readings tell us where the sodium spike that initiates the contraction is, and when overlaid with the impedance reading, can also inform where the relaxation and repolarization begins. This is important downstream as we begin looking at safety. It's important to compare both the impedance readings as well as the field potential readings to see where we're seeing effects or defects. So as we compared these different technologies in-house and several external technologies as well, including tissue bath capability as well as 3D contracture device, we found that no one technology meets all of the needs. We kind of laid it out in a table form, what we wanted on the left-hand side. Those are our parameters. And then we looked at what each system can do in our in-house evaluation and in our external evaluations. And we found that as compared to the myocyte assay, the ACEA cardio-ECR actually provides most of the information, or actually all of the same information that we can get from our primary myocyte assay, in addition to readings of hypertrophy and viability. And the throughput is much better. So all of these factors weighed in to our bringing this device in-house. I wanted to talk a little bit about the workflow that we use to assess compound effects in the cardio-ECR system. For us, it begins with taking a look at the time course of all treatments as compared to our vehicle controls. This allows us to pick an appropriate time frame to assess the dose-dependent effects and the activity of the compounds. From the field potential readings, we then get an objective look at any potential effects on the sodium spike and T wave, which can be indicators of pro-arrhythmic effects. Further analysis can also be performed from the data captured by the cardio-ECR to further evaluate mechanisms impacting contraction and relaxation, which I won't get into today. Our first step utilizes the cardio analysis template, which allows us to generate normalized data. Using this template, we can generate percent change compared to baseline readings over the course of the entire experiment, from pre-drug addition through post-drug addiction, and then further through washout of the drug. This data can be normalized to either the vehicle control or to a time point or to both, giving us a double normalization. this is what the time course looks like for contraction inhibitors using this cardio analysis template. As you can see, and here we've tested lidocaine, which is a anesthetic, as well as an antiarrhythmic drug, and we tested also a myosin inhibitor. And what you can see is that both lidocaine and the myosin inhibitor caused an immediate reduction in contraction amplitude. So contraction amplitude is graphed on the left side of each graph and the time point along the bottom of each graph. And with lidocaine, you can see that we just bottom out at all of the concentrations that we tested, and it's immediate in the effect. It's also immediate with the myosin inhibitor, but we were able to generate a good dose response with this. What looking at the time course does for us is it gives us an ability to pick where we generate our dose response. For the myosin inhibitor, the dose response can actually be generated from any one of those time points. And we can also look at what happens with the contraction after we wash out the drug as well. So this is looking further out into this same study where we washed out the drug at about 41 minutes, and then we continue to assess. And what you can see here is that you have a return of contraction. It starts to recover. This is a dose response curve for the myosin inhibitor. The IC50 that we obtained from these studies is consistent with what we observed in our primary adult myocyte assay. Here we had an IC50 of 9.7 to the minus 8, times 10 to the minus 8. And in all of the compounds that we've tested so far in this particular, all of the inhibitors that we've looked at, our results are consistent with what we see in our primary myocyte assay. An overlay of the two waveforms is shown at the bottom. And what you can see here is that when we compare the pre-drug in red to the post-drug addition in green, you can see that repression of the contraction amplitude. Additionally, you can also see that there is a slight decrease after drug addition, decrease in the beating rate, or a reduction in the beat period as well. This is also consistent with what we see in the primary myocyte assay. While it's straightforward to assess the effects of inhibitors with the iPSC-derived myocytes in the ACEA system, MyoKardia is also very interested in developing activators of contraction as well. So I want to talk a minute about the utility of iPSCs to look at activators of contraction. iPSC-derived myocytes have great potential in that they're able to beat spontaneously, have cardiac-specific responses and expression patterns, and certainly respond to negative ionotropes and arrhythmic compounds in phenotypically relevant ways. But the concerns have been raised that these cells more closely resemble fetal rather than adult myocytes, and that the cells do not exhibit well-organized sarcomeres and may have immature calcium handling, as well as perhaps not the correct ratio of alpha versus beta myosin. They also lack responses to positive ionotropes. To address some of these concerns, in the last year, ACEA has developed an activity-dependent maturation protocol utilizing the ability of the cardio-ECR to spontaneously pace the cells. The protocol that they shared with us involves exposing the cells to increasing frequency of pacing from 1 Hertz to 1.5 to 2 Hertz with 3 to 5 days at each pace. With this protocol, the cells actually become responsive to positive ionotropes. We've utilized this protocol in-house and found that we saw good responses to the beta-adrenergic receptor agonist, isoproterenol, as well as a myosin activator. This is what the time courses for these compounds look like. And as you can see, if you compare these two time courses for these two compounds, in contrast to what we saw with inhibitors, these activators have very different time courses. which makes it important to visualize the entire time course prior to choosing a time point for evaluation of activity. Then when we went to look at our dose responses, what we found is that with isoproterenol, we had a very good induction of contraction, although our dose response was a bit too high. The lowest concentration shows an increase of greater than 1.5-fold for contraction amplitude. You can also see the effects in the overlays of the traces. You can see prior to drug addition in red and post-drug addition in green. So we couldn't actually generate an IC50 in this particular experiment because what we need to do is drop our dose response. But this also is consistent with what we see in myocytes, where if we go a little bit too high with the isoproterenol, or in fact any of our activators, we see a repression of contraction. What you can also see in the traces on the bottom panel is that with the addition of isoproterenol, you see that induction of contraction, but you also see a shortening of the beat rate, or the beat rate increases. The beat period decreases, which indicates that what we're seeing is an acceleration of contraction as well as an acceleration of relaxation, which we also see in our primary myocytes. And this is what our dose response looks like for our myosin activator. It shows an excellent induction of contraction of amplitude of almost 2-fold. The IC50 generated from this compound is also consistent with our myocyte assay. And also, the highest dose that's chosen here, as I mentioned before, does repress the contraction amplitude. If you look at the overlays of the traces, you can also see that here you can easily detect the amplitude change in contraction of prior to drug addition in red to after drug addition in green. You also see that there is a lengthening of the beat period, a decrease or an increase in the beating time. This is also consistent with what we see in the primary myocyte assay. So all of this is very encouraging as we're comparing these different technologies and really wanting to generate data consistent with our myocytes so that we can reduce the amount of compounds going into this assay, as well as, or perhaps even replace the assay. So in assessing the safety of the compounds in the field potential readings, we have also looked at compounds that we know have hERG channel or sodium channel effects, as well as compounds that we know are clean for pro-arrhythmic effects, such as this myosin inhibitor. This was the inhibitor that was shown earlier in the presentation. When the field potential waves are overlaid, you can see that there's no difference in the sodium spike or the field potential duration. When we've looked at pro-arrhythmic compounds as well as compounds that we know have off-target effects, we do see a delay in field potential as well as some effect on the sodium spike. So in conclusion, we feel that the cardio-ECR and the utility of the myocytes derived from iPSCs is enhanced our understanding of what we can do in drug discovery right now. We can measure responses to both inhibitors in unpaced cells as well as activators in paced cells. We can measure all of the parameters that we can in our adult myocyte assay, which includes the contraction amplitude, contraction and relaxation rates and velocities, as well as these off-target effects and effects on the hERG channel, as well as looking at hypertrophy and viability, which I also did not show in this presentation. The format allows for a significantly higher throughput than the myocyte assay, and while the field potential also allows us a first pass at a readout on safety of our compounds as well. That's not our primary use of this equipment, but it's an important readout for us for all of our drug discovery efforts. So I'd like to thank our team here at MyoKardia, particularly our cell biology team, which includes Hong Lan, Chris, Bernardo, Jane, Sarah, and Alexandria, and my boss, Robert McDowell, who's supported all of these development efforts, as well as our IT support. Without this support from IT, we wouldn't have all of the access that we need to the software. I also have to mention ACEA's support in this endeavor. They've been extremely helpful working with us in working out protocols as well as helping to enhance the software. Thank you very much.
Thank you, Kristina, for your presentation. Our next speaker is Dr. Armando Lagrutta, who is currently Director of Cellular Electrophysiology and Pharmacology in the Safety and Exploratory Pharmacology Department at Merck Research Laboratories. Dr. Lagrutta will discuss the use of iCell-Cardiomyocytes and the Excelligence, cardio-ECR, and cardio platforms to gain mechanistic and structural insights into an adverse cardiac drug-drug interaction. Armando?
Thank you. I need control. Thank you very much. So the specific interaction that we're going to be discussing today, it was a clinical interaction between a hepatitis C virus, NS5B pronucleotide inhibitor, namely sofosbuvir, and the antiarrhythmic amiodarone. And the outline of my presentation is going to be providing a little bit of a background and rationale for the studies, and then quickly moving into the methods and results with a focus on the in vitro studies that we conducted using the human-induced pluripotent stem cell-derived cardiomyocytes, the iCell-Cardiomyocytes, and then the mechanistic insights that we gained from those studies, and also the studies in which we elucidated the importance of chemical structure features, the distereochemistry specificity, and the role of the prodrug. So by way of background, I should mention that direct-acting antivirals, which are divided in classes defined by mechanisms of action and targets, have heralded a new era of chronic hepatitis C treatment. The hepatitis C virus NS5B inhibitors, or HCV and I for short, target viral replication, specifically the viral RNA polymerase. And the sofosbuvir example is an example of a nucleoside or nucleotide analog that has been developed as a successful HCV and I. However, there's also information in the literature specifying that the chemical structure of the nucleoside or the nucleotide analogs has impact on toxicity, leading to, for example, competitor program discontinuation. So at the end of 2014, with post-marketing reports of an adverse clinical interaction implicating the drug sofosbuvir and the antiarrhythmic amiodarone started to emerge, it raised concerns that were potentially impacting all HCV and I programs, including ours. The last point of the rationale for the studies is that sofosbuvir and our own MK-3682 development candidate are prodrugs. That means they're activated in the liver in a way that involves sequential hydrolytic steps, ester and phosphoramidite cleavages, and then consecutive phosphorylation steps. So it's important to understand which molecule in the pathway is involved, what features of that molecule are relevant. And as I explained before, because these are distereoisomeric molecules, it is also important to know what role the stereochemistry plays in the drug-drug interaction. So the studies that I'm going to be referring to today were, as a full integrated preclinical assessment, including many in vitro studies and in vivo studies. But today I will focus specifically on the studies that we conducted using the cardiomyocytes and the Excelligence cardio and cardio-ECR platforms. The one thing that I should mention also is that drug-drug interactions traditionally, have to do with pharmacokinetic situations where you have the concentration of one drug impacting the concentration of the other. Early on, it was concluded that this is not the case. So it's a novel drug-drug interaction, and so the importance and of the studies that we conducted are going to be highlighted here. So the first question is, can sofosbuvir post-marketing clinical findings be replicated in the preclinical in vitro setting? And so the technologies that we use, as I explained, and very similar to what Kristina mentioned, we use the Excelligence RTCA Cardio and the RTCA Cardio ECR platforms. We also use the Cardiomyocytes. These are very robust induced pluripotent stem cells, which we can keep for greater than 7 days if needed. But what I'm showing here on the right-hand side is you can see that using the cardio platform, you can measure the cell index or the baseline impedance as the syncytium is forming. And so you can see over the course of the first 40 hours or so that syncytium is forming until you get a steady state baseline impedance. And then riding on that impedance signal, there's a small fluctuation signal, which is the beating activity of the cardiomyocytes. And so, as Kristina mentioned, you can measure an amplitude, a peak amplitude, you can measure a duration, and you can measure a rate of this signal. That's shown in the middle panel. And then in addition to that these signals that you can measure in the cardio platform, in the cardio ECR, you also have two field potential electrodes that allow you measurement of ECG-like field potential signals, including a sodium spike and then the T-wave correlates. So what we saw is upon co-application of amiodarone and sofosbuvir, we quickly saw effects on the field potential and impeded signals that we could detect on the cardio ECR platform. And this slide just illustrates the representative traces of, pre-application and application after 5 hours of sofosbuvir alone, amiodarone alone, or sofosbuvir plus amiodarone. And so the basic effect, and this is after application of 10 micromolar sofosbuvir and 0.3 micromolar amiodarone, is a big reduction on the impedance amplitude, and then we also see a speeding of the signal. So you see an increase in the beating rate. However, because the impedance amplitude is decreased, the measurement of that field potential of that beating rate increase is difficult, and so we chose to use the field potential readout for exemplification of the beating rate increases. So the next slide illustrates on the right-hand panel, you can see a time course, and these are much prolonged. We go up to 18 hours in here to try to study this effect, again illustrating the robustness of the preparation and the ability of the system to continuously and non-invasively monitor these signals. So on the top graph, you see the field potential rate, and so you can see that in the presence of sofosbuvir and amiodarone co-applied, and that's the red trace, you see a large increase greater than 1.5-fold on the field potential rate. And concomitant to that increase, then you see on the bottom plot, you see a huge reduction, almost to nothing, of the impedance amplitude. The left panels then show concentration responses, these are all concentrations which were chosen from 0.3 micromolar to 30 micromolar sofosbuvir, and from 0.03 micromolar to 0.3 micromolar amiodarone, basically trying to cover the ranges of clinically relevant concentrations. So the bottom line here is that we observe a response in vitro, but this response differs from the clinical response. The clinical response was a bradycardic effect, and here what we're seeing is a dramatic increase in the beating rate. So this wasn't really a concern, but it was something to notice, and as I'll explain later, it actually helped us understand a little bit the mechanisms that are in play. The next thing that we did is we were able to extend the results that were observed with sofosbuvir with our proprietary tool compound, which we have designated as MNI1. This is a compound that is very similar to sofosbuvir, but as you can see on the structure that is shown in the circle, instead of having a fluoride in the 2' position of the ribose, it has an alkyne group. But otherwise, it has the same diastereomeric configuration. It's an L-ALA-SP configuration, an L-alanine group and an S-phosphoramidite group. And what is illustrated on the right panels is very similar concentration responses showing the large increases in the field potential rate and the large decreases in the impedance amplitude. And then on the left panels, you can see the concentration responses of MNI1 going from 1 to 3 micromolar, and then similar concentrations of amiodarone as applied previously, 0.1 and 0.3 micromolar. So then we jumped into trying to understand the potential mechanisms that were responsible for this interaction of sofosbuvir with amiodarone. As I alluded before, we've got some early hints that there's a calcium handling dynamics as potential mechanism of action. The first thing that gave us a clue is this, what we call a calcium channel block signature, which is this increase in beating rate and decrease in amplitude. And so what is illustrated, one of the things that is illustrated in this slide, you can see in the beating rate and also on the peak amplitude. And these are experiments now that were done using measuring calcium transients on an FDSS Hamamatsu platform. So these are calcium transients. So it was important to note that these were calcium transient signals that you can see on the traces that are shown on the left-hand side, on the upper side. But what you can see is that these calcium transients basically show directionally the same effects that we had detected with the cardio-ECR. You can see basically sofosbuvir alone or amiodarone alone have no effect, but then when co-applied at the concentrations of 10 micromolar sofosbuvir and 0.3 micromolar amiodarone, you see the large increase in the beating rate and the large decrease in the beating amplitude. And again, this is a measurement of calcium transient signals. What is also illustrated in here is that a calcium channel blocker such as verapamil has the same signature. So this is an important point. So in this particular platform where you have these spontaneously beating sensation, calcium channel blockers have this tachycardic effect. They have this, show this increase in the beating rate and a decrease in the amplitude of the calcium transient signal. Or if we were measuring this on the cardio-ECR, you would see an increase in the field potential rate and a decrease in the impedance amplitude. One other point that is illustrated on this slide is the effect of the major circulating metabolite of sofosbuvir. So now this is a nucleoside derivative, and what is shown in here is no effect. So this was our first probing into which moiety in the activation of this prodrug is involved. And so we have pretty much ruled out that the primary circulating metabolite has any effects. So in conclusion then, just to try to conclude in this very complicated slide, is that the lack of response to nucleoside derivatives implicate the prodrug. And then the fact that we have a response that is similar to the calcium channel blocking response in the cardiomyocytes and similarly different from the clinical response, it starts to implicate calcium handling mechanisms. Now, this slide illustrates the effect of a major component of the calcium handling in cardiomyocytes, the sarcoplasmic reticulum. And so we're looking at the effect of two sarcoplasmic reticulum calcium store modifiers. And we see the impact of the application of these two agents, ryanodine on the left panels and tapsigargan on the right panels, on the synergistic phenotype that one sees. So, so in the blue traces, you are seeing the synergistic, the dark blue traces at the very top of the field potential rate. You can see that almost almost a doubling on the background signal of MNI1 alone or amiodarone alone. And yet, when you co-apply ryanodine at 3 micromolar, then you can see this transient, decrease, and then after a few hours, 10 hours, then you can see this effect coming back, being overridden. There was no effect that we noted on the impedance amplitude when on the MNI plus amiodarone effect when you looked at the effect of ryanodine on that. And similarly, we saw with the calcium ATPase SERCA2 inhibitor, tapsigargan, we saw a similar time course decrease on the synergistic signal of MNI1 plus amiodarone, only on the field potential rate, only on the rate effects. We didn't see too much of an effect on the amplitude. On the next slide, I illustrate the effect of the sodium-calcium exchanger inhibitor C400. This is now an inhibitor of the sodium-calcium exchanger, and what you will see in here, in this case, I'm showing the field potential signal on the left-hand side and the impedance signal on the right-hand side. But what you can see is that the effect is complex. I mean, instead of having an attenuation of the drug-drug interaction, what we see is a small increase, but significant increase. The ACEMs are actually hidden on the symbols in here, so it's a pretty big increase from about 1.8 to 2.1 on the field potential rate, and yet we see an attenuation of the effect on the impedance amplitude when you co-apply C400 on top of MNI1 and amiodarone. So these are just a few of the experiments that we conducted. In combination with these experiments, we conducted additional experiments using calcium overexpressing cell lines, which I don't have time to go into here. So what I would like to do now is just to move on to the experiments that we conducted on the cardiomyocytes and the cardio-ECR and cardio platforms to establish which chemical structure features are responsible for the cardiac risk when combined with amiodarone. Specifically, we want to look at the role of the L-ALA-SP diastereochemistry configuration versus the D-ALA-RP. And then we also want to continue looking into the effect of prodrug versus activation metabolites. This slide then illustrates we basically extended the initial findings. Initially, we looked at sofosbuvir, and we looked at the MNI1 compound, which are shown here on the table. So you can see that the sofosbuvir will have a fluoride on the 2' and the R position of the ribose, and MNI1 has the alkyne group on that R position, but they are exactly the same in terms of the prodrug diastereochemistry. Both of them have an L-alanine and a S-phosphoramidite configuration. We did a direct comparison then of 3 classes. We looked at sofosbuvir, MNI1, and MNI3, which has a chloride at that position, and we compared them with their D-ALA-RP counterparts, which are designated respectively MNI2, MNI4, and then our development compound MK3682. So we were particularly interested in knowing whether the adverse drug-drug interaction would be presented by our compound. Also shown on this table then are the primary cleavage intermediate metabolite, and this is basically what happens after you remove that phenyl group there from the phosphoramidite. And at the bottom then of the table, the NTP. So studies that we conducted with the cardiomyocytes, SYNCISIA, and the cardio-ECR helped us identify the stereoisomeric specificity of the drug-drug interaction and the critical drug of the prodrug versus metabolites. And I will illustrate that on the next few slides. So on this slide, then, we're just looking only at the field potential rate. And so in bar graphs, you can see the steady state effects after 5 hours of going from left to right, sofosbuvir, MNI1 and MNI3. So these are the L-ALA SP prodrugs, which basically have the fluoride, the alkyne, or the chloride at the 2' position of the ribose. And you can see that when co-applied with amiodarone at the concentrations that are illustrated on by the symbols in there, going from, in the case of sofosbuvir, for example, from 3.7 to 33 micromolar, in MNI1 from 0.3 to 10 micromolar, and in MNI3 the same, 0.3 to 10 micromolar. You can see the same effect where you would see almost a doubling on the field potential rate. And by sharp contrast, then the D-ALA-RP distereoisomers show little or no effect, and that is shown at the bottom in D, E, and F. Similarly, when we looked at the effect on the amplitude, then it is the same effect that I have illustrated before for sofosbuvir and MNI1 was also seen for MNI3. You see these dramatic decreases in the amplitude when amiodarone is co-applied with these agents, but we see little or no effect when the D-ALA-RP prodrugs are co-applied. Finally, in an effort to understand whether the prodrug or any of the activation metabolites were involved, we conducted an experiment in which we were looking at the PK/PD relationships. That is, we were looking at the same time that we were looking at the time course of the effect in this case, the experiment was conducted on the cardio platform. So we're basically looking at impedance amplitude, and we're looking at beating rate as a measure of impedance. So this is not a field potential. We're actually measuring it at the impedance in A. But again, it's a very similar time course. Over the course of 18 hours, you see an increase and then quickly settling a steady state after 4 hours or so for both of these effects. You see the huge increase, concentration-dependent increase whenever sofosbuvir and amiodarone are co-applied. And then in sharp contrast, with the green symbols, you see no effect when MNI2 or amiodarone are co-applied. Now, at the same time that we were doing these experiments, then we were taking samples which would be analyzed to establish the concentration of the prodrug, and this is illustrated in panel B on the right side. You can see the intracellular concentration of prodrug at the top, the primary cleavage metabolite, or the NTP. So, to make the long story short, we saw little or no formation of NTPs in the cardiomyocytes. We saw little or no formation of primary cleavage metabolite for the MNI2. However, for the compound sofosbuvir, in which we could clearly establish the presence of cleavage metabolite, over the course of the first 4 hours, you can see them, going from below the level of detection to, greater than 70 picomoles per 10 to the 4th cells. Basically what we can see is that the time course, clearly we're ruling out the NTP because the time course that it takes for the NTP to arrive doesn't match the time course of our phenomenology that is detected with the cardio-ECR. And similarly, even for the case of the primary cleavage metabolite, what we're seeing, if anything, is a slight inhibition on the initial response with the time course similar to the appearance of the primary cleavage metabolites, which is illustrated on the middle panel in B. So, in conclusion, then, what we were interpreting these data to say is that the effect versus the metabolite or NTP formation clearly seem to implicate the prodrug on the drug-drug interaction, and specifically the L-ALA-SP prodrug, with little or no effect by the D-ALA-RP prodrug. final experiment that I'm going to be presenting today, we're actually trying to address specifically the question of the effect of cleavage metabolite formation, the primary cleavage metabolite formation, by interrogating with cathepsin A inhibitors. And so these experiments, it's a little bit complicated, but I'm going to have to walk through them. What we did is we pre-incubated the CAT-A inhibitor, in this case avalactone B, for 30 minutes prior to a second addition at time 0. And we did that alone or combined with the other test agents. And so the blue symbols in here are showing, and this, unfortunately, this effect was a little bit lower. It's about 1.5 or so over the baseline. Probably has something to do with the actual concentrations that the cells saw for this particular experiment. But the bottom line is you see on the blue traces, you can see the increase in the beating rate and the decrease in the amplitude that I had been illustrated repeatedly throughout the course of the presentation. And what you can see is the application of the cathepsin A inhibitor avalactone B. You know, the only effect that we noticed was essentially an exacerbation, an enhancement of the synergistic effect of amiodarone and sofosbuvir. And this was done both by application of avalactone 3 micromolars in the gray symbols, and then pretty much superimposed on that 10 micromolar with the black symbols. And we were able to reproduce this with the MNI1, using the MNI1 as the HCV prodrug. The blue symbols, and this is shown on B, the blue symbols basically show the increase of MNI1 plus amiodarone in the field potential. At the bottom you see the decrease on the amplitude, and then you see that the effect of the application of the cathepsin A inhibitor, again, is an enhancement rather than an attenuation of the effects. So clearly the way we interpreted this was that the CAT-A inhibitory activity, which would be reducing the concentration of that primary cleavage metabolite, again, implicates the prodrug on the drug-drug interaction. So in conclusion, and I'm just going to go over all the conclusions that, including some of the conclusions that were not shown in this particular presentation. We characterized the cardiac drug-drug interaction in the iCell-cardiomyocytes, monitoring clinically relevant concentration of sofosbuvir, amiodarone alone and in combination, on signals reflecting the beating rate, in the case of the ECR, that's the field potential rate, or contractility, impedance amplitude. We show that the co-administered sofosbuvir and amiodarone induce marked increases in the beating rate and rapid pronounced decreases in impedance and calcium transient amplitudes. These effects were not observed with either agent alone or following vehicle application. So our data in the cardiomyocytes implicate inhibitor agents such as sofosbuvir or our proprietary tool compound MNI1 may interact with key intracellular calcium handling mechanisms, and those are the studies with ryanodine and tapsigargan and C400 that I illustrated. I didn't show any of the experiments that we did using the simpler test system based on influx, where we measure directly the calcium channels CAP 1.2 and CAP 1.3 in overexpressing HEC293 cells. But all of these data together suggest that these inhibitors potentiate the inhibitory action of amiodarone on L-type calcium channels. We also demonstrated the importance of both chiral centers, the L-ALA, the L-alanine, and the S-phosphoramidite moieties. We didn't show today the studies that we conducted on the mixed distereoisomers, but the bottom line is that you needed both of these chiral centers, both L-ALA and SP, to see the effects. The cardiac effects are independent of cleavage metabolites or NTP formation, and the cardiac effects of combination of L-ALA-SP with amiodarone are likely caused by the intact prodrug. Also not shown then the effects on the simpler test systems, and very importantly, especially because the signals that we detected in this system were tachycardic and not bradycardic, it was very important to show that this was translationally seen, and so we were able to show that that the emergent bradycardia was seen in an animal system, but I didn't have time to talk about that. Finally, I would like to acknowledge my collaborators on this work, on this last slide. These are all people in the cellular electrophysiology and pharmacology group in the safety and exploratory pharmacology department, along with our colleagues in vivo investigative safety pharmacology and other Merck collaborators. I apologize, looked like this thing got formatted wrong in here, but with that, I am ready to take your questions. Thank you for your attention.
Thank you, Armando, for your talk. We have now reached the end of our presentations. Thank you to both of our speakers for their talks and to ACEA Biosciences and Cellular Dynamics International for their sponsorship of this webinar. Please note that ACEA Biosciences will be hosting the 5th Annual Cardio Symposium on September 23rd in Berlin, Germany. We now have some time for our presenters to answer and discuss selected questions from the audience.
At this time, we would like to take your questions. As a reminder, to ask a question using your web console, locate the green Q&A icon in the lower left-hand corner of your screen. Type your question in the open area and click Submit.
Hi, Kristina.
Hi, Patrick.
The first question is for you. Do you use commercial cardiomyocyte cells, or did you generate cardiomyocytes in-house and use?
We actually have purchased the cells from multiple different vendors as we're doing a cell line analysis or vendor analysis right now. So all of the cell lines that we've used in our experiments are commercially acquired. And I do believe that this system would work with multiple different cell types as well.
Great. And then a follow-up question to that. Will the impedance measuring system work in other cell systems, for example, HL1 cardiomyocyte cell lines? Have you ever tested that possibility?
I have not tested cardiomyocyte cell lines, but my use with the ACEA indicates that it will work with cell lines with iPS-derived myocytes as well as primary myocytes. So I believe that, yes, it will work just fine.
Great. So the other question is, can you comment about the inversion of the twitch with chronic stimulation?
Yeah, that's a really good question. This is something that we have noticed as well. And it's not so much with the primary or with the constant stimulation. It's so over time, as we do our pacing for these cells, the waveform does not change. We don't see the inversion until we add compounds, and this is for both paced and unpaced cells. My feeling about that is that these cells may not, the waveform may change because if we're increasing amplitude of contraction, they may not have enough time to relax before the next beat. Also, with inhibition of contraction, it may be that they're just not having enough energy to actually complete the next beat. So it's the waveform changes with drug addition, not so much with the pacing.
Great. Next question then is, are the ACEA inotrope assays post long-term pacing maturation protocol performed on paced or spontaneously beating cells? And if the latter, how long after the pacing is stopped are the cells treated with compounds?
The pacing is stopped about 7 hours prior to drug addition. All of the experiments when the data is acquired are done without pacing.
Okay. Is it possible to calculate IC50 values for compounds by the cardio ECR?
Yeah, that's really easy actually. Within the cardio template or the ECR template, you can easily generate dose responses just by selecting a particular compound, and then at a particular time point, the software will do the calculation for you.
All right, and then the next question is also for you. To base cardiomyocytes continuously for days in specific hertz, how do you use the acquisition software?
You can still acquire data on impedance. You can't use the field potential readings while you're pacing the cells, but during the entire it's more like 10 to 15-day pacing. When you're doing the long-term pacing, you're acquiring impedance data the entire time.
Okay. And another question for you is, what was the cell density you used for the measurements?
The cell density for the CDI cells is typically 50,000. We have played around with cell densities a little bit. Anywhere from 30 to 50,000 cells per well seems to work very well.
All right. Armando, the next question is for you. How long can one use your platform to study long-term drug effects? Merck only used 20 hours of recording.
Yeah, for these particular experiments, we basically illustrated 20 hours, but we have done independent experiments on other projects where we can basically measure for at least 7 days. It can go for weeks. The only thing is the cells have to be fed, and the media has to be exchanged about every 2 to 3 days. But other than that interruption, then the plates can be put back in the platform, and the recording can be continued for as long as several weeks. It's not really a limitation. It's just that in this particular case, the steady state response was relatively quick.
Great, thank you. Well, that is all the time we have for questions. I want to thank all of you who joined us today for this Biotechniques webinar, and we hope that it has been informative and useful. I'd like to once again thank our speakers for their presentations, and ACEA Biosciences and Cellular Dynamics International for their sponsorship of this webinar. Goodbye.
Thanks to all our participants for joining us today. We hope you found this webcast presentation informative. This concludes our webcast. You may now disconnect. Have a great day.